Welcome back to another session of RNA biology, and we were studying about ribozymes, and this was the slide which we left in the previous class, and it is talking about the ribonuclease P, which is found in all cells which require the tRNA, 5S RNA, etc., and also the signal recognition particle. So what is the role of ribonuclease P is basically allow the maturation of this premature tRNA, premature 5S rRNA, and also premature signal recognition particle which has to undergo maturation process. And this contains two domain structure, that is substrate recognition region and also ribosome's active site. And the substrate is nothing but the premature tRNA or premature 5S RNA, etc. So this has to be acted upon by ribonuclease P for the RNA molecule to undergo maturation. So this is something which is very important for us to understand about the ribonuclease P. And the structural predictions of the RNA are made by doing phylogenetic comparisons. Now we are going on to the second group that falls under the metalloribosome category. The first category was ribonuclease P, and the second one is self-splicing introns. So normally the self-splicing intron, which is a metalloribosome, this need to have more than 200 nucleotide as the intron. Intron, like we already mentioned in the previous class, that is the intervening sequences present in an RNA which is not useful for coding purpose or not useful in the mature RNA. So introns have got their own importance and significance, etc., which we will discuss as the classes progress. But time being, you understand that when an RNA is being formed, it has regions that are need to be present in the mature form and regions that need not be present in the mature form. So those regions which are need not be present in the mature form are called as introns. And for self-splicing intron, you need to have them roughly 200 or more in nucleotides in length, and then they will be able to self-splice or splice out themselves without any intervention. In bacteria as well as in eukaryotes, the pre-mRNA or pre-RNA of the protozoan tetrahymena, the primary transcripts of the mitochondrial genes of yeast and plants all undergoes a self-splicing process, and which is governed or which is conducted by the self-splicing introns. One more thing, that this intron itself is the ribozyme. Okay, so when you talk about self-splicing ribozyme or self-splicing intron, no other molecule come from outside. It is basically the same intron fold in such a way that it can get itself out of the RNA structure. So that is called self-splicing introns or self-splicing ribozymes. Let us quickly look into what is splicing or how splicing is important. So introns by definition are segments of non-coding RNA that are interspersed among the regions of mRNA that code for protein, which we usually refer to as exons. So if a region in the mRNA that is capable of coding for amino acid, we call it as exon, and region which do not code, we call it as intron. And normally the introns are located in between various exons. So prior to translations, introns must be removed to form a mature mRNA. Let us look into this cartoon. Genomic DNA, it has got a promoter region and some untranslated region, and then exon 1, exon 2, exon 3. And in between exon 1 and exon 2, you have intron 1. In between exon 2 and exon 3, you have intron 2. And then after exon 3, you have intron 3. So while transcription, all this exon and intron are present in the RNA, and we call it as pre-mRNA. That is, it is premature, and then it undergoes so-called splicing, where exon 1, exon 2, exon 3 are spliced together to form a mature mRNA, as you can see here in the bottom, whereas the intron 1, intron 2 and intron 3 are removed. They are no more part of the mature mRNA. So this is the basic principle of splicing, and now we were talking about the self-splicing introns. So say if the intron 1 was self-splicing, don't think that intron 1 always will be self-splicing. Let us think about a situation where a given intron is self-splicing intron, then it can form a structure unique so that the exon 1 and exon 2 can come together and the intron will just disappear from the pre-mRNA. So that's what self-splicing means. So what is the difference or similarity between self-splicing and the actual splicing? Unlike common introns, self-splicing introns can splice themselves out of the pre-mRNA without the need of spliceosome. Remember, the normal splicing requires an extensive splicing machinery, which we call it as spliceosomal machinery, which is not required for the splicing of self-splicing introns. And it is basically complex of spliceosome is a complex of RNA and protein and enzymes such as helicases. They are present in the spliceosomal machinery. Although self-splicing introns can remove themselves from the RNA in the absence of any protein in vitro, in many cases in vivo, in reality, inside a cell. In vitro basically mean a lab condition in a test tube. In vivo means within the organism. So inside the real scenario. So that is in vivo. So in many cases in vivo, self-splicing proceeds in the presence of certain proteins that increase the efficiency of splicing. So you can understand if a kid or a schoolboy is there with his friends, he will be playing and can be naughty. But in presence of his teacher or presence of parent, he will behave quite sober and quite decent. Although teacher is not saying anything, although parents are not saying anything, they are not actively controlling him. Mere presence is changing the way they behave. Something similar to that in vivo, presence of certain proteins in the vicinity can enhance the efficiency of splicing. So it could be because of stabilizing the structure of the RNA. These proteins can allow a proper conformation of the RNA that could contribute to the efficient splicing of the self-splicing intron. So the self-splicing introns mediate only one round of RNA processing, unlike the protein enzyme. If you look into any protein enzyme, it can do several rounds. Say you have got, all of you know, in your saliva you have got the enzyme salivary amylase. So salivary amylase act on to starch and give rise to glucose. That is why when you chew chapati or rice for some time, after some time it will become sweet. Chapati is not sweet to start with, but you keep chewing, it will become sweeter because the salivary amylase acted on it. As the time passes, the same salivary amylase can act more and more starch molecule and convert them into glucose. But that do not happen in the case of self-splicing intron, simply because the function it is doing is self-splicing. Once it is spliced out of this pre-mRNA, it cannot do that function again. Like if you passed the intermediate exam, you cannot pass it again. You already passed. You can study for graduation, something like that. However, this removed portion of the intron can participate other biological reaction, not the splicing related, but that is also been shown. So let us see how group 1 and group 2 introns are functioning. Group 1 intron, what happens? It recruits a guanosine into one of the branches of the intron. So this line what you are seeing is an intron. What the black box and this black box are exon 1 and exon 2. So this recruited a extra guanosine, and this guanosine have got two free hydroxyl group. One is in the 2' and another is in the 3' carbon. Both of them have got a hydroxyl group. In this case, the 3' OH, remember, it is not part of this intron. It is a recruited guanosine from the outside. So this 3' OH participate in a nucleophilic attack onto the 5' end of splicing, that is towards the end of first exon. It participates in a nucleophilic attack. As a result, the exon 1 and the intron 1 junction is broken. It is no more connected here. And then what happens? The exon 1 now have got a free OH. Earlier it was not there. 3' OH was part of this double helix, part of this RNA long chain, that is part of the phosphodiester backbone. But once this nucleophilic attack happened, you will have free OH, that is from the first exon, exon 1. This 3' OH does a second nucleophilic attack onto the 5' end of the intron, that is in the 5' end of 3' end of the intron and the 5' end of the exon 2. So the second nucleophilic attack happens from the exon 1 onto the 5' end of the second exon, and as a result this intron will be released from that 3' end also. 5' end was released because of the nucleophilic attack from the G, recruited G, and the second end of the intron is released because of the 3' OH from the exon 1, and as a result the intron is released completely and you have exon 1 and exon 2 fused together. And coming to group 2 introns, it is also very much similar to that of group 1 intron, except that the group 2 introns have got a endogenous A. Instead of G, it has got an endogenous A, which is not externally recruited. It is present in the intron sequence itself, and we call it as an internal adenosine. And this internal adenosines, 3' is already part of this phosphodiester backbone. It is not available for any reaction, whereas the 2' OH is free. So this 2' carbons hydroxyl group participate a similar nucleophilic attack onto the 5' end of this intron, releasing the 5' end of the intron from the exon and now freeing the 3' OH of the exon 1. And this 3' OH undergoes second nucleophilic attack onto the 5' end of exon number 2, fusing or splicing the exon 1 and exon 2, and you end up getting a intron lariat that will be released out. So group 1 and group 2 introns does similar job. Only difference is group 1 require a external guanosine and whose 3' OH participates in the trans-esterification or nucleophilic attack reaction, whereas in group 2 intron it is an internal adenosine that participates and whose 2' OH is participating in the nucleophilic attack and the trans-esterification reactions. Same thing is whatever we discussed is listed in text. I will not go through the details because this you can read it because whatever we discussed is typed down for easy understanding for the audience. So group 1 introns are acting as the real enzymes. Self-splicing introns mediate only one round of RNA processing, unlike the protein enzyme which we already discussed. But once the group 1 intron has been spliced out, it can act as a real enzyme other than the splicing function. It can repeatedly recognize a complementary sequence of another RNA molecule by a region called internal guide sequence or IGS that can attack it by 3' OH of the bound G nucleotide. Remember, this spliced out group 1 intron is holding a externally recruited guanosine whose 3' OH is always available for attack, nucleophilic attack, and it can catalyze the degradation of various target RNA. Let us see how it is doing in detail. See, this is a target RNA and we call it as a RNA substrate, what is in the topmost panel. And now this is a spliced out group 1 intron. It has got a external G attached onto it whose 3' OH is also available for the biological function. Now, it has a unique sequence called IGS, internal guide sequence, which can pair onto the RNA substrate at a selective region. Once this pairing happened, what will happen? This guanosine can do same nucleophilic attack onto the specific regions of this RNA, similar to that what it did onto the 3' end of the exon number 1, or whichever sequence which is available in the vicinity in this case. So as a result, what happened? The RNA substrate now gets cleaved because this 3' OH of the external guanosine can attack onto the substrate RNA and breaking this RNA into 2 pieces. Now this group 1 intron ribozyme will be released and you end up getting 2 pieces of the substrate RNA. And this is something very interesting because the IGS sequence can be tweaked. So you can design the IGS sequence as you wish. If you have an RNA which you wanted to disturb or disrupt, say a person is having an HIV RNA in his body, so you can design a IGS containing ribozyme and deliver into that patient so that the IGS will selectively recognize the HIV RNA and it can cleave it. So this has got medical relevance. This is how you can make use of a ribozyme for clinical intervention. So if you look the importance of the G recruitment, the extensive secondary structure, you can see here the intronic sequence, self-splicing, intron 1 starts from here. You can see a stem loop and again a long line, again stem loop, and it is continuing, and remember it is pairing with a long distance after. So it is going up again, forming 2 loops and 2 stems, and again continuing, and then after a long distance it is again pairing. So it has got different arms, P1, P2, P3, P4, different arms are there, which is very important for stabilizing the structure of this ribozyme. And then it is this place where the guanosine is recruited, and this specific location allows the movement of this guanosine, because guanosine got recruited doesn't mean that it can perform the task. It is the structure. All these loops are important for the proper structure to be formed in the 3-dimensional structure. Then this 3' OH of the G can participate in the trans-esterification reaction, nucleophilic attack can happen. So some potential therapeutic use of the artificially made group 1 introns are listed here. We can in vitro change the IGS, internal guide sequence, and thus generate tailor-made ribozymes, that is ribonucleases, that can cleave, that is destroy mRNA molecules of our choice, like I told you about the HIV RNA. And it is a candidate method for human therapy. Currently, one such achievement has happened is synthetic ribozyme that destroys mRNA encoding the receptor for vascular endothelial growth factor. In short form, it is called as VEGF. VEGF is an important molecule. VEGF is a ligand or a growth factor, binds onto its receptor and creates the blood vasculature. You know, when cancer is developing, it needs faster supply of blood. So when you have a tumor, then inside of the tumor need adequate oxygen, adequate glucose, nutrition, etc. But when you are rapidly dividing, blood vessels need not form. So these blood vessels require, or the formation of blood vessels require VEGF. So if you target VEGF or VEGF receptor, then you can prevent the growth of cancer. So what is done is that you can create a VEGF receptor targeting ribozyme, and it's a major stimulant for angiogenesis. The formation of blood vessels is called angiogenesis. During our development, it is very important. When an organism is developed from the zygote, it is very important. But in case of cancer, angiogenesis is not a friendly thing, because the blood vessels now supply nutrition for a non-useful or a destructive cause. So you don't want such damage happening, and the blocking of its action or the VEGF receptor, if you block it by degrading its RNA, it can starve the cancer cells of their blood supply and can cause the retardation or the degradation of the tumor bulk or tumor size. So these are some of the clinical approach that is done using artificially created IGS sequence onto the ribozyme. Now coming to the group 2, so we have seen metalloribozyme, two of them, ribonuclease P and the cell splicing intron, and then comes the group 2, that is small ribozyme. Ribozymes, basically these ones are normally the viroids and the satellites. So they are of three major types. One is hammerhead ribozyme, hairpin ribozyme and hepatitis delta virus ribozyme. So these are all the major groups of ribozyme that come under small ribozyme because they are shorter in structure. What are satellites? Satellites are small RNA viruses or RNA molecules, and their multiplication depends on the mechanism of a host cell and on the co-infection of a host cell with a helper virus. Normally, satellites are harmless sequence. They do not do anything much, but they will wait for another infection to occur. Then it becomes a dangerous situation. It can enhance its effective way of attacking a host. We will see the example how hepatitis delta virus and hepatitis B virus is HDV and HBV is interacting. And ribozyme is a part of larger RNA, and we call it as viroid or satellite. Why it is called viroid? Because it is harmful sequence. However, it is not having a existence of its own. It requires another pathogen virus to enter into the host. So ribosome is a part of larger RNA that is being replicated by host RNA polymerases. So the product of the replication is being self-cleft, that is by the ribozyme activity. Say you have a sequence that is being produced by a rolling circle mechanism. It needs to be broken at frequent intervals so that it will have the meaning. That is what is being shown here. You have a RNA sequence during virus replication, and this blue line is what a newly formed strand, and it is continuing and finishing one circle. And now this strand is being replaced by the newly formed strand. So it will replace this strand which is now fallen out of this rolling circle. Now this strand need to be cut at frequent interval, without which it won't be able to make a meaningful and these pieces now it will circle and give rise to the old individual copy. And this cleaving at frequent intervals is happening because of a ribozyme action encoded in this structure. Like you may have seen a lizard shedding its tail. If a wall lizard is intimidated, we call it as autotomy. So it is breaking without any external influence. Of course, its mechanism is not. There was, I think, a few months ago, there was a elegant paper published in the journal Science about it, how the protein structure contribute. It's like almost like a ball and socket joint. If you just detach it, it just falls off. And the lizard tail is made of multiple such segments, and you can break wherever it want. A lizard can break wherever it want by simply unlocking it. So lock gets unlocked and it falls off. Same way, that is a self-action. Same way, these RNA also broken at frequent intervals. So this is a molecular structure of the RNA hairpin, and it has a backbone and also it has got unique bases in the interior, and both are important for structure and function point of view, because the hairpin, when you are talking, it has to form in a unique fashion, not any random fashion. Without a proper structure, it will not be able to perform the task, no matter which is the task you are talking about, whether it is a cleaving at frequent intervals or it is assisting a specific region to be expelled from an RNA. Everything is important for the, everything depends on the structure of the RNA hairpin. If you look further into the hairpin ribozyme, many plant virus and satellite RNA mediate the rolling circle replication. It has got two main helical regions, and one of them is called G8. G8, the guanosine 8, is essential, but its role in catalysis may be purely structural. This may be true for various metal ions, which we discussed, like many times magnesium and manganese, such divalent cations kind of stabilize the ribozyme structure. Same way, G8 in the hairpin ribozyme stabilize the various stems, like you can see stem A, stem B, stem C, and stem D. But some regions, specific bases, allow the stability, structural stability to the ribozyme. Like you can see here a region called G8, and which allow the hairpin to be stabilized such a way that the ribozyme is able to perform the task. So we will stop here, and in the next class we will continue with the hammerhead ribozyme, and we will learn more about the ribozyme and its functions. Thank you.